Back to another session of biochemistry classes. Today we'll start off with another new topic, which is molecular biology. Okay, so before we go on to what is molecular biology, you should also know certain terminologies. The first terminology that you have to know is what is genomics. So, genomics is a study of the gene and its action, and it also involves the analysis of the sequence of the DNA in a particular organism, and it also plays and it gives us information of the function of all the genes and their interrelationship. So it also identifies by which the gene combination, the growth and the development of a particular organism. So when you know about genomic, you should also know what is proteomics. This is nothing but complement of the genome. So it is a sum of the proteins that are expressed by the genome of an organism. So the study of protein properties on a large scale is to obtain a global integration view of many disease processes in the cellular process and also as a network at the protein level. So by this, what happens? We can identify the database in the cell under the normal and the abnormal conditions, and you can also analyze the or identify the defect in any particular molecular disease. So this is about proteomics. Then you have something which we call it as beta bolomics. Okay, so what is metabolomics means? It is the systematic study of all the chemical processes, especially concerning with all the metabolites, and it provides the characteristics chemical fingerprints that specific cellular processes yields. Okay, by means of this study you can get to know the small molecular metabolite profile. So some of the metabolites are the substrates or the intermediates of a particular metabolic process. So this can be analyzed by certain techniques, especially the mass spectrometry, okay, and it is used to identify and to quantitate the metabolites. And also this process, when we get the metabolites, we can separate it by gas chromatography or HPLC, high lipid chromatography, or also by capillary electrophoresis. Okay, so how do you do this? We'll just have a flow chart. The sample is collected, then the sample is prepared, and then it is analyzed by mass spectroscopy or by NMR, okay, that is nuclear magnetic resonance, and then you obtain the data acquisition and analyze and interpret. Okay, so this is the metabolics. So what do we just have a look? So you have the genomic, that is a study of the gene, and then you have the transcriptomics, that is the technology which deals with the sum of its RNA transcripts which is produced by the genome, and then you have the proteomics and the metabolite study, that is metabolomics. Okay, so this is about just an introduction about molecular biology. So next we'll go on to the cell cycle itself. So you would have studied about cell cycle in your PO classes. So just a brief description about what is cell cycle. It refers to the events occurring during the period between two mitotic divisions. So you have G1 phase, that is between the gap 1 we say, and then you have the S phase or the synthesis phase, and then there is G2 where you have the second gap and the mitosis phase. So the cell division takes place in the M phase, and it is the shortest, lasting for an hour. Then you have the daughter cells enter in either into the G0, which is the undividing or the dormant phase, or what happens, it also remains in this. You can, most of the normal cell population, you have this G0 phase, and most of the metabolic events take place during this phase. Okay, so next we'll go on to what are the different phases of the cell cycle. So as we said, it is G1, then you have the S phase, and then you have the G2 phase, or the next you have is the M phase. So in G1, it is nothing but the growth phase or the gap 1 here, the cellular growth preceding a DNA synthesis, the cells that have stopped cycling, that is recycling or the formation of the cells has stopped. So such cells are muscle and the nerve cells, and this phase lasts for around 6 to 12 hours. And then you have the S phase or the synthetic phase. There is a start of the starts up at the DNA synthesis, that is the replication. And end of this phase, each chromosome has doubled its DNA content and composed into identical sister chromatins, which is linked at the center by the centromeres, that it lasts for 8 to 12 10 hours. And then you have the G2 or the gap 2. This is a period after the DNA synthesis, and the DNA is checked for errors before the cell division. And then you have the M phase, which is the shortest phase, and it lasts for an hour. Here the nuclear and the cell division takes place. Okay. So when we talk about cell cycle, you should also know how this cell cycle is regulated. So this cell cycle is regulated at 2 points, that is you have G1 to S phase, that is checkpoint 1, and then you have G2 to M phase. So the cell damage and the repair are checked before it enters into the next phase. So you have cyclins and cyclin dependent kinases, that is CDK, at least 10, that regulates the cell cycle. So the cyclins are the regulatory units and the CDK are catalytic units. So the G1 is a checkpoint, is monitored by internal and external environment. Okay. So the G1 is mediated by a retinoblastoma protein, or we call it as ARB protein. So it binds to the transcription factor E2F and prevents it from participating in transcription. So what happens here? The RB inhibits the cell cycle at the G1 phase. So the control cells cycle, cyclin D, inactivates the RB, which is separated from the E2F, that is the transcription factor. So the tumor antigens derived from viruses like herpes virus or the human papilloma virus may combine with the retinoblastoma protein, and retinoblastoma protein cannot inhibit cell cycle, leading to continuous cell division and cancer. Okay, so the next you have another regulation by P53 oncosuppressor protein, a protein with 53 kD in size, 393 amino acids are present. It inhibits cell division and leading to cell repair. So the damage is extensive and the repair is not possible. Then what happens here? The oncosuppressor protein directs the cell to apoptosis, that is programmed cell death. So we'll just have a look at the cell cycle. So this is the cell cycle. So this M phase, that is the mitosis, where the nuclear and the cell division takes place. It lasts for an hour, enters into the G1 phase, that's for RNA and protein synthesis. It lasts for about 6 to 12 hours. Here you have the first checkpoint. The cell crossing this point is committed to the S phase. Okay, so when it enters into the S phase, it is the DNA duplication and proteins and RNA synthesis are also synthesized. This lasts for about 8 to 10 hours. Then it enters into the G2 phase. Here there is no DNA synthesis. There's only RNA and protein synthesis which takes place continuously. Here again there is another checkpoint which we call it as the G2, between the G2 and the M phase. Okay, and then it enters into the mitosis. Okay, so this is about the cell cycle. So now when we finish about cell cycle, the next topic that we have to know about is replication. Okay, so now what under replication you should know what is the definition, what are the different stages or the steps of replication. Okay, and then you should also know what are the inhibitors of replication, and also you should know the difference between the eukaryotic and the prokaryotic replication. Okay, so first we'll go on to the Definition. So what is the definition? It is a process in which the DNA copies itself to produce identical daughter molecule of the DNA. So the important characteristic of replication is it's bidirectional, that is both the directions the DNA is synthesized, and it is semi-conservative. So semi-conservative means one half of the parental DNA is present along with the newly synthesized DNA strand, right? So what is it? It is bidirectional and semi-conservative. That is, one half of the parental DNA is along with the newly synthesized DNA strand. So the eukaryotic replication begins at multiple sites. The prokaryotic, it has got only one single site or the origin of replication. So now what are the components of replication? So what is there's a list of things that are required for the replication. So the first one is helicases. Then you have the single-stranded DNA binding proteins. You have the topoisomerases, DNA polymerases, RNA primer. Then you have the deoxy, ATP, GTP, CTP, and the TTP. And then you have the DNA ligases. So these are all the components of replication. Okay. So in this picture you can see this is the prokaryotic replication and this is the eukaryotic replication. So there is only single origin of replication, whereas here in the eukaryotic there is multiple origin of replication. So you can see here the local opening of the double helix. Here you can see the opening, and then you can see there is formation of the bulb, which leads to the formation of the replication fork. And we said that it is replication is bidirectional, right? So it is both the directions you can see here. So this is in the eukaryotic where there is bidirectional replication or the formation of the new DNA. So this is the eukaryotic replication and this is the prokaryotic replication. So first we'll go on to the initiation of replication. So what we said, there is an origin of replication. It is a short sequence of base pairs which is rich in A and T base pairs. Okay, as the two strands of DNA unwinds and separates, it forms a replication fork bulb and then the fork. Okay, so what is it? It is separated by helicases, right? And it unwinds the helicase, that is the DNA strands, and SSB, that is single stranded binding proteins, keeps the strands separate. And what is the other enzyme that is required? That is the topoisomerases. It prevents the supercoiling and stress of the DNA. So there is another enzyme, that is DNA polymerase enzyme, that catalyzes the synthesis of DNA. So to initiate the synthesis of DNA, you require RNA primer, that is consisting of 10 nucleotides, is required for the synthesis of the new DNA. Okay, so this is the initiation of replication. So in the next slide you can see the diagram here. This is the two DNA strands, that one is starting from 3 prime to 5 prime and the other is in the opposite direction, 3 prime to 5 prime. Helicases, DNA helicase unwinds the double helix DNA strand, right? So this is the direction of movement of the replication fork. And then you can see the single stranded DNA binding proteins that keeps both the DNA strands separate. So this helicases, as it unwinds the DNA strand, it requires one molecule of ATP that gets converted to ATP plus PI. So this is the initiation of replication, right? So next we'll go on to what happens further. Next, what happens? The DNA polymerase enzyme can copy the DNA template in the 3 prime, 5 prime direction and produce a newly synthesized strand in the 5 prime to the 3 prime direction, that is in the opposite direction. So here you require the energy factors, that is the deoxy ATP, GTP, TTP and CTP are required, and the DNA polymerase 3 catalyzes the elongation of the newly formed DNA chain. So it uses the 3' end hydroxyl group of the RNA primer as an acceptor of the first deoxyribonucleotide and subsequently to add the other ends, and so that it grows and forms the newly synthesized DNA strand. So the hydroxyl group of the 3' end of the DNA chain group and the RNA primer acts as an acceptor of the first deoxyribonucleotide, and this end leads to elongation of the new DNA strand. Okay, so here you can see the next slide. You can see here this is the deoxy CTP, that is the entering into the deoxyribonucleotide. Okay, this is it, and this is the RNA primer. The RNA primer, the hydroxyl free hydroxyl group of the ribosome is taken here, the DNA polymerase enzyme, and it forms the newly formed phosphodiester bond that leads to the elongation or the synthesis of the new DNA strand. Okay, so then what happens after that? The DNA parental strands, that is the template, is copied simultaneously at the replication fork, although they run in the opposite direction. So what is it? The leading strand is formed by continuous copying of the parental strand that runs at the 3', 5' end towards the replication fork. So the synthesis will be from the 5' to the 3' end of the replication fork here, the leading strand, and then the lagging strand is formed by discontinuous copying of the parental strand that runs 3' to 5' end away from the replication fork with several fragments of DNA into the fork. Okay, so there are fragments in the lagging strand and leading strand is a continuous strand. Okay, so now what we said that the origin of the replication you can see here by the replication bulb and the fork, right? So it is bidirectional, both the directions it is synthesized, right? So what did we say here? The leading strand, the lagging strand is there, leading strand and lagging strand. So the leading strand is continuous, okay, that is from the 5' end to the 3' end, right? So what is it? It moves away from the replication fork, whereas the lagging strand, that is discontinuous, it goes into the towards the replication fork. Okay, so this is the origin and the leading strand and the lagging strand. So next, what is it? After that, what happens? As more of the helices is unwound, the synthesis of the lagging strand begins from another primer. The primer is required. So the short fragments formed by this process is known as the Okazaki fragments. So the synthesis of the DNA continues till it is blocked by a stretch of an RNA primer. The RNA primer is removed by 5', 3' DNA polymerase 1 enzyme. So when it is removed, the RNA primer is removed, the gap is filled by DNA polymerase 1 enzyme, and this is sealed by another enzyme by DNA ligase enzyme. So when this DNA is synthesized, there has to be certain proofreading activity. So this proofreading activity is done by DNA polymerase 3 enzyme. Okay, so we'll just have a look at the diagram of how the whole process takes place, right? So what did we say? This is the two strands of the DNA, the parental DNA helicase. Okay, so this is 3' end and this is the 5' end, right? So this is kept unwound by the DNA helicase and also by the single stranded DNA binding protein. You can see in the both ends Then what happens? The synthesis starts from the DNA polymerase enzyme, right? So this synthesis of the new strand is into the replication fork. So this we call it as this is the newly synthesized, this we call it as a leading strand, okay, that is complementary to the parental DNA, right? One, this is the lagging strand which is discontinuous and it runs away from the replication fork. So this is the RNA primer. So these short fragments we call it as the Okazaki fragments. So this is the newly synthesized DNA strand. Okay, so what is it? This is the DNA polymerase enzyme, right? And this is the RNA primer, and this DNA polymerase 3 recognizes the RNA primer and begins to synthesize the DNA. Okay, so this is the whole process of replication. So in the next you can see another picture, the parental strands separated by DNA helicase. So what happens here? This is the RNA primer, the DNA made by DNA polymerase enzyme 3. You can see here also you have the DNA polymerase 3 enzyme. Okay, so what is it? The first one, the RNA primer is elongated by the DNA polymerase 3 until another stretch of RNA is encountered. So you can see here that is RNA primer till another RNA stretch is countered. The RNA primer is removed by DNA polymerase enzyme 1. Here you can see one ribonucleotide at a time. The gap is filled by DNA polymerase enzyme 1 and the remaining neck is sealed by DNA ligase enzyme. Okay, so this is the lagging strand and this is the leading strand. So the leading strand runs into the replication fork, whereas the lagging strand runs away from the replication fork. Okay, so when we talk about the DNA polymerase, you should also know the different types of DNA polymerase enzyme. So you have in the E. coli and in the mammalians, you have 1, 2 and 3 in the E. coli, and you have alpha, epsilon, beta, gamma and delta. So the for 1 and the alpha 1, it helps in the gap filling and synthesis of the lagging strand. Then the DNA polymerase 2 or the epsilon, it helps in the DNA proofreading and repair. The beta, it helps in the DNA repair. The gamma, mitochondrial DNA synthesis, and DNA polymerase 3, that's the delta, it helps in the processive and leading strand synthesis. Okay, so DNA polymerase enzyme can come as a 3-mark question. Then you'll have to write the different functions, the different types of DNA polymerase enzyme. So when we talk about the whole replication as such, you should also know what are the inhibitors of replication, right? So you have antibacterial agents and anti-cancer agents. So the antibacterial agents like ciprofloxacin, nalidixic acid and novobicin, they are all inhibit the bacterial DNA gyrase enzyme. Then you have the anti-cancer agents like the etopside, which inhibits human topoisomerase enzyme 2, adramycin, it also inhibits the human topoisomerase 2 enzyme. Then you have 6-mercaptopurine, it inhibits human DNA polymerase enzyme. And you have the 5-fluorouracil, it inhibits the thymidylate synthetase enzyme. Okay, so these are all the inhibitors of replication, right? So next we'll go on to what are the differences between the prokaryotic and the eukaryotic replication. So the DNA is circular in the prokaryotes, whereas it is linear in the eukaryotic. The origin of replication is in one place, whereas in eukaryotic there are multiple sites. Then the single-stranded binding protein cooperates binding to the ssDNA, and ssDNA binding protein binds at the replication fork. Then the RNA primer, it is required for the synthesis of both the strands. Here the DNA polymerase alpha has a primase activity and initiates the synthesis of both the leading and the lagging strand. Then you have the DNA polymerase enzyme. It is a major polymerizing enzyme is DNA polymerase 3. And here the major polymerizing enzyme DNA polymerase delta and DNA polymerase epsilon is important, right? The next other difference is the proofreading activity. DNA polymerase 3 also has the 3' to 5' exonuclease activity, so that any wrong base is removed and the correct one is added. Here the DNA polymerase delta and epsilon both have 3' to 5' exonuclease activity and therefore serves as a proofreading function. And then how the gaps are filled. The RNA primer is removed and the gap is filled by DNA polymerase enzyme. In the eukaryotic, the RNA primer is removed by RNA primase H and FENE1, and the polymerase beta is involved in gap filling and DNA repair. So what are the inhibitors? Inhibitors in prokaryotics are ciprofloxacin and novomycin inhibits the topoisomerase enzyme. Whereas in eukaryotics, it is epoxide, andromycin, and also you have camptothecin and inhibits the topoisomerase enzyme. So this is about the differences about prokaryotic and the eukaryotic replication. So next we'll just summarize the whole process of replication. Okay, so the origin of replication is identified. Then there is unwinding of the parental DNA to form the replication fork, right? And then the RNA primer complementary to the DNA template is synthesized by RNA primase enzyme. Then the DNA synthesis is continuous in the leading strand, that is towards the replication fork by the DNA polymerase enzyme. Then the DNA synthesis is discontinuous in the lagging strand, that is away from the replication fork, as called as Okazaki fragments. And then there is elongation in both the ends of the DNA strand. The synthesis is from the 5' to 3' direction. Then the RNA pieces are removed. The gaps are filled by deoxynucleotide by DNA polymerase enzyme, and the pieces are ligated by another enzyme, that is DNA ligase. Now the proofreading activity is done by DNA polymerase enzyme, and finally it gets organized into the chromatin. And the main enzymes involved in replication are DNA polymerase enzyme, helicases, topoisomerases, DNA primase, single-stranded binding proteins, and DNA ligase. Okay, so this is just a summary of what the whole replication process is. Okay, so this is it about replication. So replication itself can come as a long note. So you can be asked, most commonly it is the eukaryotic or the prokaryotic can be asked as the replication. You can answer this same question as the same thing and give the difference between both the eukaryotic and the prokaryotic replication in the end, right? And give a summary about the inhibitors also along with it. And please see to that you draw the diagrams also pertaining to each step from the origin and then how it gets elongated and how the whole replication fork is formed. Okay, so this finishes with replication. In the next class, we will continue with another topic of molecular biology. Thank you. Have a nice day.